have our afternoon session of graduate student talks, which will involve 2 talks about research by Joe Piechura and Nick Guia, and then 1 journal club by first-year grad student Alistair Ramirez. So Joe is going to tell us about his work on understanding how constitutive circadian regulation of transcription in cyanobacteria modulates or doesn't modulate more variable transcriptional responses to environmental stressors. So first.
Like Alicia said, my name is Joe. I'm in the G4 and Aaron O'Shea's lab, and today I want to tell you about my work looking at the interaction between stress-responsive systems and circadian systems in cyanobacteria. So broadly, I'm really interested in the general question of understanding how cells can sense and respond to their environment so they can appropriately function under a wide range of environmental conditions. And to provide my own answer for this question, I'm using cyanobacteria as a model system and trying to think about how they sense and respond to their environment. So I'll begin by introducing you to cyanobacteria. Here I'm showing you a liquid culture of the particular species of cyanobacteria that we study in the lab called Synechococcus elongatus. And if you take some of this culture and put it under a microscope, you can indeed see that these are single-celled bacteria with a rod-like shape. And something you'll also likely notice about this culture is its beautiful blue-green color. And that's because cyanobacteria are photoautotrophs that produce these colorful pigments that allow them to harvest energy from ambient sunlight to produce ATP and reducing power. So as organisms that rely almost entirely on available sunlight to produce energy, cyanobacteria will face very predictable challenges. And that's because, as we all know, we're on a globe that's rotating around an axis in a very periodic manner, and this rotation will essentially remove the source of energy for these organisms every single day. So to prepare themselves for these predictable changes in energy availability, cyanobacteria use something called a circadian clock. And a circadian clock is an oscillatory system that produces rhythmic changes in activity with a period of roughly 24 hours. And these changes in activity essentially serve as an internal representation of the time of day for an organism. And what's particularly amazing about these systems is that they'll continue to produce oscillatory changes even in the absence of changes in environmental conditions, which really demonstrates that these are autonomous oscillators within cells that are serving as essentially a clock. So to actually use this timing information to produce changes in activity, cyanobacteria produce rhythmic phosphorylation of a transcription factor called RPAA. So if we grow cyanobacterial cells under constant light conditions and then measure RPA phosphorylation over time, here I'm showing you time in continuous light in hours and the percent of RPA that's phosphorylated, you see that when cells have been in constant light for 24 hours when they expect it to be dawn, RPAA is present at a low level of phosphorylation, and its phosphorylation will increase as the day goes on, reaching a maximum when cells expect it to be dusk at 36 hours in constant light. And RPA phosphorylation will continue to show this oscillatory pattern of phosphorylation with a period of 24 hours if light intensity does not change. And the phosphorylation of RPA allows it to associate with DNA to regulate gene expression. So by controlling the phosphorylation of RPA, cyanobacteria can use their circadian clock to regulate gene expression. As photoautotrophs, cyanobacteria will also face very unpredictable challenges. For example, you can measure the amount of instant sunlight in a field as was done in this paper. Here you can see time in hours on the x-axis and photosynthetic active radiation on the y-axis, which is a measure of light intensity. And what you notice about this graph is that there are very short timescale changes in light intensity that occur throughout the day. Here you notice that light intensity decreased for a period of time and then increased again. And these events are occurring because of things like transient shading, like a cloud passing between the sun and the earth. And as organisms that are trying to harvest energy from the light that they're receiving, these changes in light intensity will alter the metabolism of these organisms. And they can even induce stressful conditions by producing things like reactive oxygen species. So it's no surprise that cyanobacteria have a system that allows them to sense changes in light intensity so they can respond appropriately. So you can grow cyanobacterial cells under low light intensity conditions and then rapidly transfer them to an environment with a higher light intensity. And what you find is that the phosphorylation state of a transcription factor called RPAB will rapidly decrease. So here I'm showing you time in high light, and here I'm showing you the amount of phosphorylated RPAB. And you find that when cells are in low light at time 0, RPAB is present at a high level of phosphorylation. And as soon as you put cells in high light, the phosphorylation state of RPAB decreases. And similar to RPAA, the phosphorylation of RPAB allows it to associate with DNA to regulate gene expression. So by controlling RPAB phosphorylation in response to changes in light intensity, cyanobacteria can regulate gene expression in response to these stimuli. So to recap, cyanobacteria use a circadian clock to control RPAA to regulate gene expression. And they also have signal transduction systems that can sense changes in light intensity to control RPAB, which can also control gene expression. And if you think about it, it becomes apparent that environmental conditions will be changing constantly while the circadian clock is active in cells. So these systems will be operating at the same time in cells almost every day. And because of this, we think this is a particularly useful system to try and understand how circadian and environmental stress response systems might interact. So to begin to understand how these systems might interact, we asked how the expression of single genes might be regulated by the circadian clock or by changes in light intensity. So to determine if a gene shows a circadian or light-responsive gene expression pattern, we used RNA sequencing to measure the abundance of RNAs under varying conditions where we expected the activity of these systems to vary. So to determine if a gene is potentially regulated by the circadian clock, we measure the abundance of transcripts over 1 day under constant light conditions. And if the abundance of this transcript will vary under these conditions, we can hypothesize that it's regulated by the circadian clock. And to determine if a gene shows a light-responsive gene expression pattern, we measure the abundance of RNAs in cells grown under low light intensity, and then again when these cells have been rapidly transferred to a high light intensity environment. And we did this at a time of day when RPAA would be active later in the day. So if the abundance of a transcript changes when light intensity rapidly increases, we can hypothesize that light-responsive systems regulate its expression. So when we do these experiments, we can identify genes that show a circadian gene expression response but don't show light-responsive gene expression. And one such example is this gene of unknown function called 2267. So if we measure the abundance of this transcript over 1 day in constant light, we find that this transcript when cells expect it to be dawn is present at a very low level, and the abundance of this transcript will increase throughout the day, reaching maximum when cells expect it to be dusk, showing a very clear oscillatory gene expression pattern. So because of this, we can hypothesize that this gene is regulated by the circadian clock. However, if we measure the abundance of this transcript when we rapidly increase light intensity, we find that when cells are in low light, this transcript is present at a high level, and when we transfer cells into high light, the abundance decreases a little bit, but it really is maintained at a high level, suggesting that this gene is not strongly regulated by light-responsive pathways. So conversely, we can identify genes that show light-responsive gene expression patterns, but not circadian gene expression patterns. And one example is this photosynthesis gene, PSAD. So if we measure the abundance of this RNA when light intensity varies, we find under low light conditions that the gene is very highly expressed, but when cells are transferred to high light, the abundance of this RNA rapidly decreases, suggesting that this gene is somehow regulated by light-responsive pathways. But if we measure the abundance of this RNA over 1 day in constant light, we find that it's maintained at a constant high level, suggesting that this gene is not regulated by the circadian clock. And finally, we can find some genes that show both circadian and light-responsive gene expression patterns. And one example is this gene RPOD5. So if we measure the abundance of this RNA over 1 day, we see a very clear oscillatory gene expression pattern with minimal expression at dawn and maximal expression at dusk, suggesting again that this gene is regulated by the circadian clock. And if we vary light intensity, we find under low light conditions that it's highly expressed, and as soon as cells are put in high light, the abundance of this transcript decreases, suggesting that this gene is regulated also by light-responsive gene expression, or light-responsive systems. So to summarize this part, we find that the circadian clock and light-responsive pathways seem to be able to regulate gene expression independently at some genes, but at other genes, the two pathways seem to be co-regulating gene expression and converging. So we were interested in understanding a bit more how these genes are regulated mechanistically. And since we know that RPAA and RPAB are central regulators of these pathways, we wanted to ask whether any of these genes are directly regulated by RPAA or RPAB. And to do this, we used ChIP-seq under varying conditions to determine where in cyanobacterial genomes RPAA and RPAB are associating under different conditions. So briefly, ChIP-seq is a method that tells you the abundance of different sequences that can be immunoprecipitated with your protein of interest from cells. So this data is displayed as normalized read counts, which are a measure of the abundance of different sequences as a function of chromosomal position. And I'll be showing you the position of genes of interest as gray boxes on these plots. And ChIP-seq data for RPAA will be in red, that for RPAB in blue, and that for a mock experiment where we use antibodies that shouldn't bind anything of interest is in black. So if our protein of interest is associating with a promoter, what we expect to see is an increase in read counts above the mock data at a specific position. So from this data, we can be pretty sure that RPAA is associating upstream with this gene. So to determine if RPAA regulates gene expression, we carried out ChIP-seq under constant light conditions at different times of day. So we did this at dawn when RPAA is present in a lowly phosphorylated state, and then again later in the day when RPAA becomes phosphorylated. So our expectation if RPAA is regulating a gene is that it should associate later in the afternoon when it's phosphorylated, because phosphorylation is thought to allow these proteins to associate with DNA. And to determine if RPAB regulates gene expression, we did ChIP-seq under conditions where light intensity varied. So we did this under low light conditions when RPAB is phosphorylated, and then again when we've rapidly transferred these cells to a high light condition when RPAB becomes dephosphorylated. So our expectation here is that RPAB should associate with promoters under the low light condition when it's phosphorylated. So first we can ask how this gene 2267 is regulated, which we saw had a circadian gene expression response but was not light responsive. And if we carry out ChIP-seq under constant light conditions, and we do this experiment at dawn, we find that neither RPAA nor RPAB seem to associate upstream of the open reading frame of this gene. But if we do this experiment later in the day when RPAA becomes phosphorylated, we see that there's a significant association of RPAA directly upstream of this gene, correlating very well with when this protein becomes phosphorylated. And if you remember earlier, this gene shows very low expression at dawn and much higher expression in the afternoon, which correlates very well with the association of RPAA upstream of this gene. So because of this, we can propose a model where RPAA, when phosphorylated, will associate with this promoter to activate the expression of this gene. And because of this, this gene will show circadian gene expression responses because it's directly regulated by RPAA. But since this gene does not show light responsive gene expression responses, we can be pretty sure that RPAA can act independently of changes in light intensity at least some genes. So next we can ask how this gene PSAD is regulated, which was light responsive but didn't show circadian gene expression. So if we carry out ChIP-seq under conditions where light intensity varies, under low light conditions we detect significant association of RPAB with the promoter of this gene, suggesting that RPAB is regulating its expression. And if we repeat this experiment when cells have been transferred to a high light environment, we see that the association of RPAB decreases significantly in concert with the loss of phosphorylation of this protein. And if you remember from earlier, this gene is highly expressed under low light conditions and lowly expressed in high light, correlating again very well with the association of RPAB upstream of this gene. So because of this, we can propose a model where phosphorylated RPAB can associate with this promoter to activate gene expression, which allows this gene to be light responsive. And again, since this gene doesn't show circadian gene expression, this seems like RPAB can act independently of the clock in some genes. And finally, we can ask how this gene RPOD5 is regulated, which showed both circadian and light responsive gene expression. So we can carry out ChIP-seq under constant light conditions at dawn and in the afternoon. And what we find is that RPAB is associated upstream with this gene at all times of day. But we find that RPAA will only associate upstream with this gene later in the day when it becomes phosphorylated. And if because of this, we can hypothesize that both RPAA and RPAB are directly regulating the expression of this gene. And if you remember from earlier, this gene is not expressed at dawn when only RPAB is associated with the promoter. So because of this, it seems like at this promoter, the association of RPAB with DNA is not sufficient on its own to activate gene expression. And next, we can repeat these experiments when we vary light intensity. So again, under low light conditions, we detect the association of both A and B upstream of this gene. But when we transfer cells to high light, we find that both of these proteins dissociate. And if you remember, this gene is highly expressed in low light and lowly expressed in high light, which is correlating very well with the association of both of these proteins with DNA. So here, it seems as though this promoter is acting as an AND gate, where it will only be activated if both RPAA and RPAB are phosphorylated and associated with the promoter. And because of this, this gene will show both a circadian and a light responsive gene expression response. So in summary, we find that circadian and stress responsive systems can control transcription factors to regulate gene expression independently in some promoters. But in other promoters, it seems as though the information encoded in both of these proteins is integrated such that both of them are required to activate gene expression. And because of this, these genes can show variable gene expression that are dependent on both of these systems. We find it very mechanistically interesting that these proteins seem to be able to activate gene expression by themselves at some promoters, but at other promoters they seem to require both proteins in order to activate gene expression. And we're really interested in using in vitro biochemistry and reconstitution to understand how these proteins are mechanistically activating gene expression at these different promoters, to see how cells can use the same pieces of information differently at different genes. So with that, I want to thank my boss, Aaron, for all her support and guidance, and my lab for being really good friends and scientific mentors, and also Catherine for hearing practice talks, and my committee, Andrew, Mike, and Vlad for all their guidance. And with that, I'm happy to take any questions. So they normally bind DNA as dimers, so the phosphorylation induces their dimerization, and then they can bind a bipartite motif, and we can identify those. And I can find places in the genome where I can show in vitro that both of these proteins will bind directly. But interestingly, for example, at both of these genes, I can't detect that these proteins directly bind DNA. So there seem to be evidence that they can activate gene expression in the absence of directly touching DNA, but maybe indirectly by associating with polymerase. So basically they can bind sequences, but they can also seem to be able to regulate gene expression in other ways. Andrew?
Different light in the afternoon, is that some stressful or toxic level of light that they should be doing things to avoid, or that sort of normal idea?
No, like the very abrupt increase in light intensity will produce reactive oxygen species, which will destroy photosystem complexes. So they induce a lot of chaperones, and they induce new photosynthetic apparati that are more resistant to light intensity. They'll also downregulate some photosystem genes. So yeah, it's a very stressful condition. But
it's a high intensity relative to what they would see in their real world, and sort of like the clouds suddenly going away and the sun coming in.
Yeah, so I guess, so the perturbation I used initially was just a rapid increase in light intensity, because we know from the literature that this will activate this pathway, but more natural light would show, like a very gradual change in light intensity over the course of the day, which could allow them to acclimate and respond to things differently. And I guess instead of a very rapid increase in light intensity, more realistic perturbation would be a rapid decrease and then an increase. So our future experiments, we're interested in seeing how these pathways will act under more natural light conditions to see how they might, work under more relevant conditions. Yep.
Do you think that just with YouTube protein, you can have that sort of high-pass filter that filter out with very high variation that you can see? Or do you need to have another system upstream that will actually do the high-pass filtering prior to regulating all the genes?
So it's, I guess, on a gene like this one that's only regulated by the circadian clock would probably, I guess it has a built-in high-pass filter because the circadian clock itself is not affected by these rapid changes in light intensity, at least as far as we know. Oh. So, filtering out rapid changes in light intensity? No, oh, okay. But yeah, so.
So basically, it's just that you're going to, again, are you trying to see if you can make a single little solution that will do the faster pretty much correct?
I haven't done anything like that, no. So,
if one of them is that and is not actually
yeah, so I've done some preliminary looking at so I can detect that RPAB directly binds DNA at the sequence directly below this peak, but I don't find that RPAA associates directly with DNA, even if I add it together with RPAB. What's particularly interesting about some of these peaks is that they reside directly over what we think the transcription start site is. So because of that, my hypothesis is that this peak actually represents RPA touching RNA polymerase. So presumably we could try in vitro experiments mixing these things together, seeing if they will bind, if they will activate transcription. Yeah, that's a really good question. Like, I feel pretty, like, that's a really good question. So, the next thing I would probably do is see if I can assemble RNA polymerase in the presence of RPA only at a promoter like this, and then from there, try to understand more about how they could select specific promoters. Yeah. Yeah, so they definitely do bind DNA at some places, but yeah, that's a good experiment. I haven't done anything like that.